Swayam Prabha. Digital India. Educated India.
Dear students and participants, so in this lecture we'll discuss what is the next step or what are the next steps required for further progressing of this technology, like the organ printing technology, to actually realize the potential of this technology, that is producing artificial tissues and organs. Right? We have seen developed. We have been discussing actually the various aspects of this technology, starting from what is 3D bioprinting, what are the different types of 3D bioprinting techniques exist, also what are the strength and limitation of this different technology, what are the critical parameters of these different technologies, and also how we can use that technology for different applications. Then also we have discussed like several biomaterials concept, like biowings, what are the different types of biowings, single material, multi-material biowings, what are the different typical physiochemical characteristics or features of this biowing, that also there are the limitation and challenges with these biowings. Then we have discussed, uh, we have then started what is required and how to print in tissue and organ, and also how, suppose we are targeting a particular tissue construct, tissue and organ constructs, what is required to print that particular tissue and organs. We have discussed a few tissues, not we have not few tissue and organs, we have not covered all these things because we don't have, because there is a time limitation also, so we have not covered, we have not gone into the details of different 3D printing of different other different tissue organ structures. But I have given some hints about printing of at least kidney, heart, liver, musculature, cornea, all these different tissues, organ structures. So now, and then also we have discussed novel techniques like 4D bioprinting and in situ bioprinting. So in this lecture, what we'll do, this lecture is mostly like a summary of the whole course, like whatever we have discussed in this, whatever we have discussed till now, we'll try to again summarize all these different points, and then we from there will extend that to what is required to take this technology to the next level, like the focus of future research, what are the focuses, different focus, what are the focus of future research, right, focuses of future for future researchers. So because many of you are probably now interested to work in this particular discipline, in this particular like in organ printing, and probably thinking what is the best project or topic that I can work on. So here I can give you a list of things that can be, that is the need for to develop further, and so that if you are interested you can work on a particular topic. I have typically listed these the focuses like this, the points into as per their area, like bioprinting technology, biomaterials, cell source, and also like vascularization, innervation, and maturation. So we'll start with the bioprinting technology first. So in bioprinting technology, we have discussed various types of bioprinting technology like extrusion-based bioprinting, inkjet-based bioprinting, laser-assisted bioprinting, digital light projection bioprinting. So all these different things we have discussed, and now probably you must have a better idea about how different these different bioprinting technologies work, and also the process parameters related to those bioprinting techniques, right? So most important thing for bioprinting technologies, it should be compatible with physiologically relevant materials and cells. And that is the focus of future research, where people are trying to develop different new novel materials, and also how we can use different other types of cells for bioprinting. But the most important thing is the bioprinting technology should not induce any type of toxicity or any kind of adverse effect towards these materials and cells. Like when we suppose we use a particular types of materials in a particular type of bioprinter, there should not be any change or alteration of that properties, physical properties of that materials. Even when you make a structures with that material, there should not be any deleterious effect of these materials, or there should not be introduction of some kind of deleterious things, or like it suppose any kind of infection, any kind of contamination, that should not be included within this thing, within the materials and cells. And also it should not cause toxicity to the cells, like the cell viability, as I earlier mentioned, cell viability is of prime importance. So that's why the bioprinting technology should not be causing any cell damage, and the cell viability should be very high with this printing. So it should be that's why the first point, it should be compatible with physiologically relevant materials and cells, and there should not be any change into that status of these materials and cells, right? So that you should understand that, and there is a huge body of research actually how we can actually further progress or further modify this technology to suit a particular to suit printing a particular type of cells and particular type of materials. And there are also, so one can also think of suitable projects into that particular thing, how we can actually improve the materials aspect and the cell viability in particular in by any particular type of bioprinting techniques. The next thing is the increased resolution and speed, because we have seen till now, we have seen that the resolution of bioprinting is varied. Like inkjet printing has a very high resolution, and also extrusion based bioprinting has a lower resolution. But when we actually discussed on organ printing, there we have mentioned, or I have mentioned that the organ are actually typically they have micro to macro structure, and also the internal architecture of these organs are also have nano to micro architecture. So to recreate or reproduce that kind of architecture or that hierarchy, resolution is the key here. And if we can improve the resolution of that bioprinting system, it can actually help in understanding, or it can actually help to solve that problem of creating very fine intricate microstructure that can be done with this increased resolution. So resolution can be improved, and to increase the resolution, also in case of extrusion based bioprinting, there also we have discussed that the because the resolution of extrusion based bioprinting is directly dependent on the nozzle diameter. So nozzle diameter, we cannot reduce the nozzle diameter to very low, because actually basically the, the cells while they are encapsulated in a biwing, they should be passing through that nozzle, right? So that nozzle diameter, we cannot keep the nozzle diameter very close to the cellular dimension, like 20-30 micron if the cellular dimension is. So nozzle diameter, suppose if you keep the nozzle diameter 50 micron, it can probably the resolution will be better, but then it can lead to decrease in cell viability, because the cells will be under tremendous shear stress passing through that narrow gap. So that's why the resolution, so that's what we have to think, how can we improve the resolution. For laser assisted bioprinter, that is not the case. There actually we can work on better resolution thing. The other thing comes the speed. As you have now understood that while during printing the cells are in a condition where that is not very good for sustaining for longer time. So that's why we need to increase the speed of the bioprinting techniques, so that we can come up with, or we can print very, we can print the structure very fast, like this, so that the fabrication time can be reduced, because the fabrication time has some effect on the printed structures, because if the fabrication speed is good, then the time required is good, then the cells will be healthy in the printed structure. Then they can be provided nutrients, like the media, they can be cultured under the optimum conditions. But during printing, that thing can be taken care of. So the speed of the system has to be optimized without compromising the cell viability and cellular and the resolution of the printed structures. So that though we have to increase the resolution, we have to also improve the speed of the structure. Again, these two are contradictory, because suppose if we, our resolution is very high where we can print suppose very fine structures, then also that can lead to delay in printing process. So there we have to come up with a better, like we have to come up with some technology where the resolution can be better along with the speed. Then the scale up for commercial application, and in many during many of the lectures I have stressed on this point, how the scaling up is on major limitation for this organ printing. So how we can increase the scale of this bioprint or the, how we can increase scaling up, the process can be faster, or the we can print a like functional tissue or organ that is of sizable functional tissue and organs, like liver is like that kind of big structure can be fabricated. That can be typically done by, as I earlier also mentioned, by mini tissues or modular concept, where you can actually print different tissue structure, different models, and you can make them, connect them together, and then you can actually come up with this thing. Because for commercial applications, when actually suppose this technology when actually will go to clinic for treating patients, that time scale up is the will be the major bottleneck. If suppose you cannot print a big structure, then scale up will be the major issue. So that has to be taken care of. Next point is the combining bioprinting technology to overcome technical challenges. Here also we have discussed this also earlier, that a hybrid bioprinting approach, like where, because we know very well, suppose this laser assisted bioprinting can, the resolution can be very high, but extrusion based bioprinting actually can be used to develop a centimeter size of tissue constructs. Now suppose an inkjet, with inkjet bioprinting we can print very, we can pattern very well, right? We can, because it's basically spraying, creating droplets, and the droplets can pattern where cells can also be loaded, and the droplets can be patterned on a certain. So now how can we can we combine different bioprinting technologies to overcome the technical challenges? So how that can be done, that is another major issue. And there are some research groups, I can say, that working on that, these aspects, where actually they are combining both. Suppose they are combining melt extrusion based 3D printing along with inkjet or maybe laser assisted bioprinting, or maybe some people are working on, suppose, electrospinning plus extrusion based bioprinting. So combining different bioprinting technologies along with the other technology also can help us to overcome the technical challenges, because with a single printing technology it is very difficult to print micro to macro and also nano to micro. So this kind of different scales, different architectures printing is very challenging, and a single bioprinting technology can is very difficult to, with for a single bioprinting technology can to do that. So that's why combining or hybrid bioprinting technology can be very well used to develop, to overcome these technical challenges. So for bioprinting technology, these are the challenges, and these are also the focus for future research, and anybody can work on these things. And there are actually some groups are actually working on these different aspects of this technology, and to overcome the challenges of the associated with this bioprinting technology. Now the next point is we're going to discuss is biomaterials. So it is also mostly like you can consider the bio wings, in our case in organ printing. So in terms of biomaterials, There are requirements like complex combinations or gradients to achieve desired functional, mechanical, and supportive properties. Like there are few, and there are some cases like where we have seen that because the tissue or organ structure is so complex, they because most of the times, if you consider the ECM also, ECM is a combination of diverse various or varieties of bioactive factors, bioactive molecules like there are carbohydrates, there are proteins, there are growth factors, different other bioactive factors. All these things are there. So to achieve, because of that, sometimes this ECM, the combination, the different ECM molecules, they are present in different ratios, different order, and also in different gradients to add. So that's why they, because by this way they only support even the cellular also, sometimes the cells are present also uh, present in a gradient manner, so that a particular function or even that function can be a desired function or can mechanical support structure can be possible. So well, by printing also we have to come up with technologies or strategies by which how can we, suppose how can we pattern biomaterials, how can I use different types of biomaterials within a structure, within this printed structures, how can I generate gradient, how can I generate different types of architectures with different biomaterials. Also, can you use a different complex a combination of biomaterials to see whether they can actually mimic that chemical composition of that particular tissues or organ structures, so so that we can able to generate different functional mechanical different supportive and mechanical functions by doing that. So this is the actually where we have to use this biomimicry approach. I have discussed earlier that biomimicry approach can be very well used for doing that, and that has to be done for this thing. Other this thing, this can be also modified or designed to facilitate deposition while also exhibiting desired post-printing properties. Like during bioprinting, we can actually modify or design certain process to the so that facilitate the deposition of the biomaterials in certain way. Like suppose we want to create a hierarchy or a gradient, so that deposition process can actually help us to do that. But so that we can actually come up with a desired functions, post printing properties can be very well be tuned, so that the suppose here 40 bioprinting can also come into play, but where we in 40 bioprinting, as we have discussed earlier, where we use this process where we can print the structure in one particular 3D configuration, then but it after post printing upon when the stimulus is activated, that time the structure can changes itself to another 3D configuration. So this post printing property, suppose if we can control the post printing property of this property by taking care of this deposition process, by taking care of the biomaterials, taking of biomaterials, then probably we can come up with better strategy, better technology to address certain limitation, to address so that we can actually address the issues that is related to the that is related to bioprinting. So this is another way to do that, and there are various groups, research groups again working on this working on this aspect, where they are coming up with, they are actually trying to reproduce the ECM structures and also ECM chemical properties. They are trying to mimic that and then try to use that for different applications. And it has been shown that when actually you can try to you can recreate that ECM, or you can mimic that ECM chemical physical chemical properties, then the cellular response also has been improved, can be enhanced further, and that is probably that is another way to deal with this thing. Other than this thing, there are various research groups. Even we in our lab also we use there is digitalized tissue specific ECM scaffold to study ECM composition and as a printable material. So what we do, we use different types of digitalized extracellular matrix. That means we collect different tissues and then we can we can use a process called dissolution, by which we can throw out all the cells. The cells are not required because those are the mostly immunogenic things. So we can throw out the cells, and then we can use that ECM material, and that ECM material is very cell supportive, and also it can direct the stem cell differentiation also can be further directed by with the help of the DCM materials. We have actually observed that in our laboratory also. So the digitalized, so that's why there are various research groups now, they have started using decellular extracellular matrix. And also we have also developed a technology by which the DCM material can be used to develop biowing. So DCM biowing also can be developed, and that can be further used further for bioprinting of different tissue structures, and in a way that can definitely help us to direct tissue growth, or to guide the tissue growth in a particular way. So that's why this in the biomaterials, these are the points that we have to deal with, and these are the various research problems can be generated and can be addressed to tackle this issue, like where somebody can work on this, how we can best recreate the ACM structures or nano or microstructures with the different types of biomaterials, how we can generate gradient, how we can generate different types of patterns, how we can modify and design the suppose the biowing to facilitate deposition, also exhibit desired post printing properties, and also how different visualized tissue constructs can be, or decellized tissues can be used to develop biowings for this thing. Next is the cell source, and cell source, if we have, when we discuss cells like the in bioprinting, we have always discussed the cell source is the primary concerns for bioprinting, because only if you don't get cells, then how can you do the bioprinting process, right? So there can be different types of cell sources, and we need to identify proper cells cell sources, so that we can have the cells. And another important thing to remember for bioprinting, we the cell requirement of cell number is very high. So how can we get that many cell numbers? That is also another thing to be discussed. And mostly the cell source should be well characterized and reproducible sources of cells required, because once we have, suppose we identify a particular cell source, then that has to be properly characterized, and also we have to develop the process by which we can get reproducible sources of cells. Otherwise there can be a variation, and that can lead to different other challenges. Many a times these are also regulatory challenges, because the regulatory body also, they are also trying to understand how good, or how, what are the different ways we can actually can be the cells can be collected, isolated, characterized, and used for bioprinting applications. So there are various types of cell source. Earlier also I have discussed various types of cells. Some are like primary cell source, stem cells, primary cells, differentiated cells. These are like different types of cell sources, and how well we can use them, that is on another thing. Stem cells are like now more and more autologous stem cells are being used for different tissue, different organ printing applications, because autologous stem cells there is a very less chance of immune rejection, and because these are patient's own cells, and then definitely these autologous stem cells can be very well used for treating any type of particular diseases. So that's why cell source we need to identify, and we need to characterize the stem cells properly. Another thing is combination of cell phenotypes with specific function. That is also another important thing, because the mostly what happens when we culture the same these primary cells outside the body in a tissue culture condition, that time they generally lose their phenotype, and it's also along with the specific function. So how can we actually preserve the phenotype? What type of techniques can be used? Suppose if we take help of certain biomaterials that can preserve the phenotype and the function of the tissue during bioprinting, that can be always can be taken care of. And the combinations of these things has to be addressed, and then only it can help us to do this thing. Next thing is the, in case of tissue or organ, in case of organ, we have seen that a heterogeneous cell types are present there, like different diverse types of cells are present there, and all these different cells, they have their own function. So that's why we need to reproduce this thing. Like suppose in case of liver, the liver cells, like the hepatocytes, they are involved in case of for metabolism. Cholangiocytes are present. Cholangiocytes cells are there. They are present. Their function is for bile circulation, bile production. Similarly, for endothelial cells, they are present in the endothelial sinusoids, but their role is to how the blood, from the blood, when the blood is going into the tissue, like the then yeah, controlling that thing is the function of the endothelial cells. So so different cells actually they perform different types of function in a particular tissue or organ. We need to understand that what are what are the different cell types present, suppose in a particular tissue and organ, and what is the function of this each of these cell types for for that thing. So now suppose if we understand that, then only we can able to reproduce that, and that can help us to generate a particular tissue structure where all the, suppose if we produce a particular tissue or organ structure where all the different types of cell types present, so then only we can able to reproduce or recreate all the different functions that a particular tissue or organ does to do. Again, can we control the cell proliferation and differentiation of those or the cells towards that particular applications? Like suppose when we use that cell source for a particular, suppose for bone tissue engineering, suppose when you are trying to bioprint a bone, then can we control the cell proliferation and differentiation within that construct? How can I do that? Can be done with some small molecules or other factors. If can we use some small molecules or other factors to actually direct the cellular function, like this, how the cells will proliferate, how they will differentiate. As I mentioned, if the same cells can be differentiated towards particular lineage, how can I control the differentiation of that stem cell toward particular tissue lineages? Can you do that? So these things also, like if we can do that, then that definitely that can help us to achieve the goal. That means we can develop particular tissue structure, organ structure. So suppose our interest is to print a piece of bone, so that can be very well done if we can understand these points. So that is in case, so in cell sources we have to come up with reproduce well characterized and reproducible sources. Combination of cell phenotypes with specific functions are very much important for understanding, for and also for to generate a particular tissue structure, tissue or organ structures. Greater understanding is required of the heterogeneous cell types present in the tissues and how we can actually reproduce them, how we can actually use different types of tissue cell types and then use them for bioprinting. That is another thing to be discussed, another point to be remembered. And how can you control the cell proliferation and differentiation by using some means, like either it can be small molecules or other factors, to come up with cell source that like sustaining and healthy cell source, that for bioprinting is very important. And how can I do that? So all these points are very important for understanding this thing, for understanding the bioprinting, the focus of this bioprinting of this, what is the next level, how we can take it to next level. So thank you for attending this lecture. In the next lecture we will discuss a few more points in the next steps in 3D bioprinting. Thank you.